Now I am going to start another important topic of the genetics, that is the linkage. And mind it, linkage and crossing over are the two faces of same coin, and at the last I will prove it. So linkage and crossing over are the two faces of same coin, remember it. In crossing over, the tendency of genes just to remain separate, but the tendency of linkage is just to bring the linked genes together. This is the basic difference between linkage and the crossing over. So the tendency of linked genes to remain together, the tendency of linked genes to remain together, First, let me explain what is the linked genes. Linked genes means all the genes present on same chromosome are called linked genes, and when they are present on same chromosome, there is least possibility of their separation. Actually, the case of linkage discovered just after the discovery of Mendel's independent assortment. And let me explain one thing, that in Mendel's test cross, we obtain 1:1:1:1 test cross ratio, but that ratio is the mathematical one, that is not the real one. The real value was 4:4:4:4. The mathematical value is 1:1:1:1. So in linkage there is variation of the Mendel's test cross ratio. The two scientists, they are Bateson and Punnett. Just try to repeat the Mendel's dihybrid cross in another plant. Another plant is sweet pea, and botanical name was Lathyrus odoratus. But they could not repeat the Mendel's result. It means their test cross ratio deviate from the Mendel's. So in this case, our problem divided into three parts. First one, we will have to see what was the experiment of Bateson and Punnett. Second one, what was the new test cross ratio, and third, why it occurs. So here I am going to divide the problem into three parts. First of all, I am going to discuss the Bateson and Punnett's experiment. This experiment was like the Mendel's dihybrid cross.